As many of us know, cancer is very insidious, afflicting over 10 million Americans. Over the past 40 years, scientists have formed a good understanding of the genetic and biochemical causes of this disease. However, we still need to construct a comprehensive framework to also take into account the physical environment in which cancer develops. To aid in this effort, researchers at Johns Hopkins University at the Institute for NanoBioTechnology are investigating the physical cues which guide tumor growth dynamics. But what exactly is cancer?
Cancer has traditionally been looked at as the uncontrolled growth of cells forming a primary tumor.
The primary tumor, however, is just the beginning of this disease. As the tumor grows, it invades the surrounding stromal space and ultimately starts affecting other parts of the body. This process, known as metastasis, is when cancer cells reach the circulatory system and get transported to distal sites to form a secondary tumor. It is this secondary tumor which kills about 90% of all cancer patients.
Angela is working on a project that is really at the core of trying to understand how this early step in this metastatic cascade is truly occurring. Is it cells invading as kind of a collective entity, still somewhat loosely attached to one another and moving into this stromal space to then encounter blood vessels and be on their way to these distal sites? Or is it cells living as singlets, one after another, maybe following tracks they may leave behind into the surrounding matrix, and then be on their way to encounter these blood vessels and invade distal organs?
To understand the early steps in the metastatic cascade, we use balls of cancer cells and grow them inside of gels made of the most abundant protein in the human body, collagen. We vary the collagen density to mimic different physiological conditions and see how this ball of cancer cells grow in these gels. We observe that different densities trigger different behaviors. For instance, balls of cancer cells growing in denser gels are constrained from movement and thus proliferate. On the other hand, less dense gels allow individual cells to break off from the main ball of cells to invade into the collagen gel. These observations are interesting because they help us relate cancer's physical environment to its behavior.
This discovery that in a way the microstructure, the physical properties of the matrix can so dramatically influence the switch from growth to go is essential, because it really proves that the physical environment plays as much as a role of what we could call cell intrinsic properties. And this interplay between what's called the microenvironment and the tumor is what Angela's project is all about.